Well, to all of you joining us on Zoom today, welcome. I understand we have lots of people who have registered for this event, so I'm going to wait just a minute or two before we get started, and then we'll take it away. All right, I'm tempted to start. I'm looking at the participants list here. I think we've already reached 80. I assume we might have a few people come and join us in a minute, but I'm tempted to make good use of our time. It's a real pleasure to welcome all of you to this MIE Research Spotlight event. My name is Marcus Bussman. I'm the professor of mechanical engineering here at the U of T, and at the moment I have the privilege of serving as chair of our Department of Mechanical and Industrial Engineering as well. And it's a great pleasure today to introduce a colleague who's going to give what I think is going to be a really amazing presentation on his work in microscale robotics. Professor Eric Diller, just a little bit of background, is an American, did his PhD at Carnegie Mellon University, and I think came to the University of Toronto. We hired him in July of 2014. Eric has quickly built a big lab around his research interests in small-scale robotics and in magnetic control of those robots, and he's going to tell us about that today. And in his first 6 years, so he's coming up on 6 years at the university, 6 or 7, has really won a number of awards that speak to his expertise and his accomplishments, both as a researcher and as a professor. And so I could list a number of them. A couple of years ago, in fact, he won the Early Career Teaching Award in our Department of Mechanical and Industrial Engineering. He's one of the students' favorite teachers, but has won a number of research awards as well. Some Canadian awards, an Ontario Researcher Award in 2017, an award from the Canadian Society for Mechanical Engineering as a young researcher. And last year won a couple of more distinguished awards for his work. Every 2 years, the Faculty of Applied Science and Engineering here at the U of T gives out a Charles Early Career Research Distinction, and Eric won that. And then a couple of months later, the good news just kept coming. He was awarded the IEEE Robotics and Automation Society Early Career Award, which is a global prize that is given to, I think last year it was given to a couple of young faculty in the area of robotics for in recognition of a great start to an academic career. I think the Department of MIE is very proud and very impressed by all that Eric Diller has accomplished so far. And with that, I'd like to have Eric take it over and give us what I think will be a really entertaining presentation.
Well, thank you, Marcus, for the kind introduction. And I'm really happy to be here. And I think this is a great kind of event. It's really nice to be able to connect with alumni, which isn't something that we have a lot of opportunities to do. So I'm excited for that. And I know that some other participants are here as well. And I'm just going to put my slides up here, which you should be able to see now. And today I'm going to tell you about some of the work that we've been doing towards medical applications of tiny robots. And in particular, we want to put robots into the body. And so today I'm going to tell you about 2 applications that we've been working towards going into the brain for doing brain surgery and going into the gut, the gastrointestinal system. So I'm going to show you how we can make small mechanical devices. And you know, I'm a mechanical engineer by training, and in my lab we take a mechanical engineering approach with to these robotic problems. And so this shown here is one of our surgical tools. I'm going to tell you about this and some other things here. Okay, so I'm going to start off by getting right into one of our motivating cases for what are we trying to do with robots and where do we think that they're going to be useful. And the first is in the brain, in removing brain tumors. And in North America, there's 25,000 malignant or growing brain tumors diagnosed per year. And this is especially an issue in children, where it's the most common form of cancer. And we're most interested in solving the difficult cases where there's tumors deep inside the brain. For example, here, which is called the pineal region, which is in the neighborhood of these purple regions of this illustration, which are called the ventricles, which are these fluid-filled cavities inside the brain. And so reaching deep into the brain and doing surgery is difficult. And in fact, surgery is often the recommended first treatment, maybe followed by chemotherapy or radiation therapy. But going in and getting in there and physically removing the tumor is often the first step. And the outlook for adults is not great with brain tumors. For children, most do survive, but they have a long road ahead of them. And the procedure for doing removal of a brain tissue through surgery is very invasive. And I'm going to show you on the next two slides some graphic pictures here of what a procedure looks like and how a surgeon can get in there. And so this picture on the top left shows sort of the plan for cutting back the skin on the skull and actually peeling it back and exposing the skull. And the white dotted regions here are regions of the skull that are actually cut out and removed. This is called a craniotomy, so that the surgeon can access the brain, push the brain tissue aside, and insert a bunch of tools in. And using a microscope, you can see a view through a microscope in the video here. So this is what a surgeon can do, and they're able to remove tumors from all different places by accessing from different angles. But this is very, very invasive procedure, and everything has to be sealed up and titanium plates added to secure the skull afterwards. And so there's a lot of interest in doing these procedures in a less invasive way. And so for decades, people have been trying to do minimally invasive or endoscopic approach, where instead of taking a huge chunk of the skull off, can we just remove a small hole? So bore a hole and insert smaller tools through the hole deep into the brain. And you see here some commercially available and clinically used endoscopic tools. So these are straight tools, they're inserted down into the brain. And some procedures can be done this way. Some brain tumors can be removed if they're located in a favorable place. They can be removed this way. But as you see in the video, the dexterity of these tools is quite limited. To move the tool side to side, the surgeon has to move the entire apparatus, which is sort of swiveling around that little borehole. And so there's a need for more dexterous tools. And in fact, the limitation here means that this procedure is not done that often, and usually a more open approach is done. And in fact, one study which I was looking at suggested that 1 in 4 of these endoscopic tumor removal procedures fails to remove the full tumor, and 1 in 5 has complications. So a dexterous set of tools is needed to improve these procedures and enable all brain tumors to be removed, or majority, through a less invasive approach. Now, you might be familiar with surgical robots. You hear it in the press, and you may know someone. For the last 20 years or so, these have been actually used in real patients, in human patients. And this is just a demonstration video of the most commonly used platform made by Intuitive Surgical, an American company. And this is their robot called the Da Vinci. And you see that these tools are fantastically dexterous. They have these little wrists, and the two hands can move together. It can tie knots, they can suture, cut tissue with scissor versions, and there's a bunch of other tools that can be put on the end of these. And this is a fantastic tool and really a marvel of mechanical engineering. You can see pulleys, and these are driven by cables. And right now you're seeing the view that the surgeon would see, which has this fantastic vision through this camera, which is also inserted next to the tools. But even though these tools are small, they're not small enough to go into the brain. And tools need to be about half the size. These ones are 5 to 8 millimeters in diameter, but to go into the brain, they need to be about half the size. And due to the mechanical complexity of these tools, we can't just keep scaling them down. You know, a lot of people are trying to do that and just use good mechanical engineering to design smaller and smaller parts and process of assembling them and making them strong and reliable. But there's a bit of a bottleneck, and going any smaller runs into a number of issues. And so we've been asking this question: how can we make a dexterous robotic surgical tool small enough to fit into the brain? It needs to be about half the size of these tools, or about 2 millimeters across for the tools. And so our solution to this, or what we've been trying to do, is rethink the approach. Let's not take a mechanical approach. Let's not run a bunch of cables through here, but can we make the tool even simpler? And so how do we drive this? And here's a video of my son pushing, or trying to grab this magnet. And of course, we're pulling the magnet around wirelessly using a magnet under the table. So he's perplexed at how this happens, but this concept, which you I'm sure have done before, this is the approach that we're trying to use. How can we use magnetic fields to move and control tiny tools inside the body? And can we do this with the dexterity of those surgical tools, those surgical robots that I showed in the video earlier? And can we do this reliably, and can we make them super tiny? And so I'm going to show you how we've been working on this. And this has been the question which has driven me for the last 10 years, and it is designing mechanisms that can move with magnets. And so I'll talk about the two applications today, in the brain and in the gut, which are two of the exciting ones that we're looking at. Okay, so magnetic fields. So this is a magnetic coil system in our lab. My students built this system, and it consists of 6 magnetic coils, and we put our experiments on the inside, and it allows us to apply magnetic fields to tiny robots that go inside. And magnetic fields have some great properties for doing control that allows us to do something pretty sophisticated, as I'll show you through the rest of the talk. So first off, magnetic fields can be projected over a distance. So this coil system is 20 or 30 centimeters in overall size, and we can easily project fields over these kinds of distances, even up to a meter over the size of the human body. We can project fields. Think of an MRI machine, which makes very large fields. So for medical applications, we can make these magnetic fields and project them into the body. The second great capability or factor of magnetic fields, which is useful for us, is that magnetic fields will penetrate through tissue. And in fact, for low frequency magnetic fields, which is what we primarily use, tissue and most materials are transparent to the magnetic fields. So as long as you don't have an implant with iron, nickel, or cobalt in it, it doesn't matter whether you put your arm in the system or there's air in the system. The magnetic fields will propagate through the system in the same way. So this is great for doing controlled tests in the labs and translating them to application. And lastly, we can create torques and forces on tiny magnets, right? That's how we're going to do our control. So we're going to attach magnets onto our tools, onto the little robots, and we're going to exert those torques and forces. And this is the only equation we're going to get into in this talk, but it's important because it captures the essence of all the rest of the control that we do. So I'm going to explain these. So we can create a torque, T, on our little magnet M. So M is like a vector pointing from south to north pole of our tiny magnet. And just like a compass needle, this magnet will try to align with a field B. So if we apply a field, a magnetic field B, it's going to pull that magnet into alignment with the field, just like a compass needle. So that's how we can apply torques and twist mechanism components or entire microrobots into different orientations. Now, at the same time, we can also apply magnetic forces, F. And so magnetic forces, we can pull things around. And this is, you've all felt this when you've played with magnets, but actually this simple, relatively simple vector expression encapsulates that entire effect. And you see this del B, this is the spatial gradient. If you think to your vector math courses, del B is the spatial gradient, or really the shape of the field. And so you can think about magnetic force as pulling magnets towards regions of higher field strength. Okay, and so to create torques and forces, we need to control the field and the shape of the field. And the good news is, using a coil system like this, we can do both of those in a precise, prescribed way. And we can actually calculate things. If we want to create a desired torque and force, we can calculate what coil currents are needed to generate that input. So this is our basis for control and leads to our approach for miniaturizing robots to be small enough to go into the body. We're going to remove all of the complex mechanisms that we can, get rid of the pulleys, get rid of the cables, and let's just deliver power and signals using that magnetic field. So the rest of this talk, I'm going to first introduce a basic microrobot, what we can do with this, how we can make a mechanism. And this is going to be a small gripper. So I'll show you how we make these. And then we're going to take that same concept and apply it to two applications, which require a little bit different version of that tool. So let's get into how can we make a small gripper, drive it with magnetic fields. And this is a cartoon of our desired gripper function. We'd like to be able to levitate the gripper, move it through 3D, open and close the arms, and go and grab objects, manipulate them in the environment, put them in a target site, something like this. So this is our ideal behavior. And so now how do we actually achieve this? And how would we drive this with a magnetic field as the only input? So this is the function or the guts of our gripper to do this. So you see here we apply a field, and as the field is applied, the gripper closes. And the gripper itself is made from these black blocks, which have magnetic material in them. And this is a permanent magnet material, so we can magnetize it in advance. And it's actually made from a rare earth magnet material. Same thing you may have seen if you ever see a magnet advertised as super strong or rare earth, it's probably made from the same material. So we cast them into these tiny 0.5 millimeter size little blocks. And then we connect them with these flexible hinges made out of silicone rubber, similar to like a spatula in the kitchen is made from a version of that same rubber. So we cast this into this shape, which involves some manual assembly. My student Jia Chen spent many hours under a microscope assembling these components. And the key to how it folds up now is shown at the bottom. How are each of the blocks magnetized? And each block, as I showed before, is going to behave like a little compass needle. So these red arrows show the magnetization. Think about south to north pole is each of those red arrows. And each arrow is going to try to align with the field. So if we apply a field vertically, the base magnet here is already aligned, so it doesn't experience any twisting torque. This first arm is going to experience a counterclockwise torque, and same thing for the other arm segments. And so the result is what's shown in the illustration. All those arms are programmed in advance to fold up. And you can imagine we could program them differently to get a different kind of deformation, which actually gives us a large design space for making mechanisms. And what's more, the magnetic field has more than just a strength. It's a vector field, and it has 3 key attributes which we can use to control different motions of our gripper. So the strength, the direction of that vector field, and the shape. And each of these controls a different thing. The strength of the field opens and closes the gripper, like I already showed. The direction of that magnetic field controls how is our gripper oriented. So the gripper is always going to align with that field. And finally, the shape of the field will pull the gripper towards regions of higher field strength. So these are actually 6, if you count them up, these are 6 different inputs. You could think about we load it into a vector with 6 desired states, and we can actually control all of these independently by controlling the coil currents that generate those fields. And so we've developed a mathematical mapping for this. And any combination of these inputs you want, we can create that. So we can do control of our micro gripper. Now, to put this into a robotic system, we need some feedback. And in the lab, for basic experiments, we just use cameras with a microscope lens on the top and side. And these give images like that's shown up at the top here. Top and side view. We use some standard computer vision techniques to extract the position of key features like grippers or cargos. And then using the principle that I just showed, we're able to control the currents in our coils and generate fields and forces to move our microrobots. And we can calculate this quickly and design a control system to do it. So my student Jia Chen did this excellent demonstration. We have a top and side view of the same experiment going here. And we have the gripper. It's going to pick up this cargo, transport it over the wall to the goal. I'll just show the video now. This is actually an autonomous experiment. So we just clicked go, and using computer vision, you see the object is tracked. The gripper microrobot is tracked. It takes 2 attempts to pick up the cargo. And we've programmed a sequence of these behaviors so that it can deliver the cargo over to the right side. So this is the basic capability. We can make a mechanism. We can get it to deform in the desired way using these flexible hinges and magnetic elements, and we can move it through the environment. And I mentioned that one issue was the assembly of this thing. So how do we assemble those components, which is quite tedious. My former master student Tianqi figured out a way of using a 3D printing approach. So he could print out these grippers in one process into any desired shape. And during the printing, he could actually program the magnetization of each of those segments. So we could get grippers with any number of arms. I asked Tianqi if he could make a walking robot. And this is the great video he came up with. This is an 8-legged walking sheet, basically. And each of those legs has a different magnetization direction. And this shows the potential, right? We can design complex behaviors. Like he could design what type of gait this thing's going to walk with when we apply different types of clever magnetic inputs. Like this thing is actually driven by a rotating magnetic field, which creates the cyclic motion of all those legs with different phase. So this is what we can do with the mechanisms. Here's Tianqi with one of those grippers, just to illustrate the size, which is a few millimeters across. In fact, we can do some other things like control multiple devices. And so this work was done by my former PhD student, Mohammed, who figured out how to make this clever control method work. Here you see 2 grippers going to pick up cargoes, and we can actually move them independently along any arbitrary paths that we want to do parallel manipulation. And the way that this works is a bit complicated, so we're not going to go into the details, but you can think about 2 grippers which have magnets on them are going to interact with each other magnetically. So they'll pull and push and orbit around each other. And so Mohammed figured out how to actually regulate that pull and push so that they'll assume any relative positions that we want, so we can do independent control. So this is what we can do. They can move it and deliver cargoes. So this is our basic capabilities. And those grippers are great demonstrations, but they suffer from a few issues. They're not very strong. They're basically just flat sheets of rubber, and they wouldn't be able to exert enough force to interact with real tissue or survive journey through the body, these kinds of things. So I'm going to show you how we adapted this same principle first into the brain. So this is our vision. We'd like to have tools which can be inserted deep into the brain, into those ventricles, the fluid-filled cavities, and deploy some grippers and scissors or other tools. We're going to control it with magnetic fields. And we'd like a surgeon to be able to drive this from some sort of console like other surgical robots. On this work we're doing with a neurosurgeon at Sick Kids, the Children's Hospital here, Jim Drake. And so looking at our concept a little closer, we're going to have an endoscope. So a camera is going to be watching this thing as it's inserted into the ventricles of the brain. Our tool is going to be deployed here, and we'll be able to open and close the grippers, and we'll have some wrist joint, which can move up and down, left, right, and open and close. So those are the basic features that we want driven by the magnetic field. Our project actually has some other collaborators, including Professor Hani Naguib in MIE department, who develops thin film sensors, which we're also trying to add to our tool to give feedback to the surgeon, although I won't get into that today. Okay, so this is our surgical tool prototype number 1. So this is basically a different version of that gripper that I showed you a few minutes ago. Instead of being a flat sheet of rubber, we've made it stronger. We make it out of metal. It has 3 magnetic segments, which are connected by springs. So these are metal springs now. And at the back end here, there's a wrist joint that allows it to bend. You see it bent, the wrist is bent in the top picture and it's straight in the bottom picture. And these gripper jaws can open or close as well. And you can see the magnets have different magnetization directions. So if we apply fields in different directions, it's going to exert torques on these magnets, which cause the wrist or the gripper jaws to be deformed. And so here's what it looks like as a video. The gripper is fixed on the right-hand side, and we're applying a field. You can see the field is that summation of the arrows that are being drawn here. And the gripper bends down, and now we're closing the jaws to pick up this tiny piece of polymer, just a plastic block here. We can grab it and move it around. So this shows the basic capability of our surgical gripper prototype number 1. And so we're going to stick this into the brain through a tube, which is inserted into the brain. My former student, Andrew, came up with a bunch of different versions of this tool, which are actually optimized in terms of magnet placement and spring design for different desired behaviors like maximum gripping strength or maximum bending angles. And so we can actually insert this, and we've tested it in our lab in this rubber brain, Phantom. So a fake brain made out of rubber, and you can see the ventricles here. And we put this rubber brain into a magnetic generating system. And this system actually uses permanent magnets, you can see there, instead of coils, but it's the same concept. Okay, so here's what it actually looks like. Our tool is inserted next to an endoscopic camera, and this is the view of that camera on the right. You can see the tool is long and thin going here with the jaws, and it's grabbed this simulated tumor. And actually, we're using a cooked pea as our simulated tumor, which is sometimes used for surgical training as simulating the mechanical properties of a tumor in the brain. And you can see here we bent the wrist to go to the site carefully, and we're able to grab that and pull it out. So this is our surgical tool prototype. And we're still working on new versions of this. And this is a second version of our surgical tool, which is still under development. So I don't have any videos for you yet of this one. You can see it up here inside the ventricles of that rubber brain. Here it is, the tool inserted next to another instrument for applying suction. And this tool, instead of having those long springs, which led to some undesirable behaviors like vibration and undesired bending, this one uses pin joints. So it's a simpler design mechanically, although it does introduce some friction. So we're still investigating whether that is a fundamental issue or whether we can do nice control even with these tiny pin joints. But this is our second version of the tool, and this one is a little bit sleeker as well. Looks like those real surgical tools. So initially I said our driving motivation is to make smaller tools. And so how big are these tools we've made? On the left here, you see these are scaled drawings. These two are the surgical robot tools that I showed in that video at the beginning. So these were the ones dissecting the grape. And these are 5 and 8 millimeters wide. And on the right are our new magnetic tools. Version 1 that I showed the video, and version 2, there's a large and a small version of this, which we're developing. And the small version is at our target size. This is our target size, about 2.5 millimeters across. Just to get a scale, because everything's so tiny, I put some peanuts here so you can see these are really tiny tools. And we're able to recreate a lot of the dexterity of those larger mechanical robotic surgical tools. Now, the last capability we'd want in our surgical tools is cutting with scissors. We need to cut tissue. And so my former PhD student, Niza, figured out how to replace those gripping fingers with blades. And she made this totally mobile version. So this is not even tethered. It's able to move on its own. And you see it basically consists of 2 blades with magnets on it. They're connected by a spring at the back. So it's kind of like a shears design. And here she directed it to cut that pillar off. And the pillar is made of agar gel, which is simulates a very soft kind of tissue as a first kind of test. So we're able to do cutting. NISA did a great job of optimizing the placement of these magnets and design of the spring so that the magnets actually pull on each other in an ideal way to aid in the cutting. And so we're working on a more sophisticated version of this scissors, which is sleeker, smaller, more controlled as a tethered or untethered tool. Okay, so the last part of the surgical application that we're working on is how do we generate the fields, right? This coil system, which is used for some of these demonstrations that I showed, this is not going to work in an operating room, right? We can't stick a head inside this system. So we need a bigger system. And in talking to surgeons, we need a system that's out of the way. So we can't surround the entire head. They need to be able to get in there. They need to be able to insert tools. They need to be able to have access to the patient. So I'll give you a quick sneak peek at some of our plans, what our vision is for how this would work. One version which we're working on is mounting a bunch of coils on a robot arm. So this would offer a very flexible approach. The magnets could be brought in as needed or pulled away. And actually the magnets could be moved around the head even during a procedure to get the optimal placement for best magnetic control. Another option, which is a little bit simpler in the environment, which my student Adam is actually building this scale system right now, is just putting the magnets underneath the table. So it's a little bit less ideal than magnets surrounding the head from a control standpoint, but we've shown and optimized this. So we've shown that it's going to be able to perform pretty well, and this is actually going to generate stronger fields for stronger motions than what I've shown in the slides already. Okay, so what's next? Otherwise, I mentioned a few times that our forces are a bit limited sometimes. They scale with the volume of magnetic material on board our microrobots. And so one way that we've been thinking of increasing the force is adding a transmission. And so my former master's student just graduated, Mihai, developed this prototype of a mechanical transmission which doesn't have any gears, has minimal frictional rubbing components. It's based on strings that twist up. And this mechanism, he was able to get many times stronger forces. This version is 60 times stronger than those previous tools. And we get gripping forces around 1 Newton in force, which we think is what we need for doing gripping and cutting inside the brain. So this is great, and we're working on planning to make smaller and sleeker versions of these transmission mechanisms. Of course, we need a control and interface, and we plan to use a similar approach to other surgical robots for doing that. And of course, we also want to add more features, add not just 1 tool, but 2 tools. And so I have some students working on this problem. My student Sajad showed that theoretically we can control 8 different inputs. So you can think about each arm has up, down, left, right, open, closed. We could control 2 of those arms using magnetic fields. So we're trying to actually demonstrate that practically. And we hope in the next year to take some of our basic tools and use them for doing real surgeries in animals as a trial. Okay, so we're going to move on to the second application, which is going into the gastrointestinal system, the gut. And one of our biggest motivating cases for doing this is for studying or diagnosing what's going on inside the small intestine. And you may be familiar or know someone who suffers from inflammatory bowel disease, which affects quite a lot of Canadians with a large treatment and management cost. And this is inflammation of the small intestine. Historically, it was thought that this is caused by diet and stress, but now it's understood that it's a more nuanced combination of factors, including, in recent years, there's been an explosion of understanding that the microbes, bacteria included, are actually contributing or maybe even causing some of these illnesses. And IBD has a strong link that people suffering from IBD have different types of microbes in their gut. And so if this connection can be solidified, if a causal relationship can be identified, that different bacteria actually cause the illness or affect it, that can inform new treatments. And these treatments can be much less invasive than for example, pharmaceutical treatments. You could take probiotics or prebiotics, which are basically like food for your bacteria. So rather than directly managing your own cells, can you manage your gut bacteria to manage your health? This is the promise. And there's an incredible amount of interest in this, but sadly, there's no great way of studying how this works. And so if we look at, try and look at inside the small intestines, what's going on in there in live patients, we can't get in there very easily. Colonoscopy can reach into the colon, the large intestine, but that's an invasive, unpleasant procedure and can't easily get into the small intestine at all. And so studies of this, people trying to figure out what's going on, typically use stool samples. So looking at feces and taking genetic sequences of the bacteria in feces and trying to make conclusions from that. But stool is not a good indicator of what's going on in the small intestine. In fact, because of some factors like decreasing oxygen concentration going throughout the gastrointestinal tract, the microbial community is quite different high up and low down in the GI tract. And if you look at the different microbes in a stool sample, which are illustrated by these colored bars here, taken from a paper on this, the bacteria population of feces bears little resemblance to which bacteria are located at regions higher up in the intestines. So just looking at feces, which is what is primarily done, doesn't give us a good story of what's going on. And so a molecular biologist who works on this, named John Parkinson, who's now my collaborator, a few years ago asked me this question: can we design a tool to sample from the small intestine directly so that we can improve our understanding of this problem and build some stronger relationships and understand what's happening? And this is the concept that we came up with together to swallow a robotic pill, which would travel through the gastrointestinal tract on its own. And at the right time, we're going to activate it. Trap door will open, take a sample, then seal up and pass the rest of the way out, and we could collect it after it's excreted. So this would be a non-invasive approach. So healthy patients could do this with only a small amount of discomfort, swallowing a large pill. We could do this and deploy it on a large scale. And then we could take samples from different places. My collaborator, John, is an expert in doing genetic sequencing of microbial samples and drawing biological conclusions from this. So figuring out from gene sequences which are expressed in the sample, which type of bacteria are present, and then using that to inform some biological or clinical decision-making and understanding. And we're not the first people to think of putting smart pills through the GI tract. In fact, for some decades, clinicians have been able to give patients a camera in a pill, and it goes through and takes a video or images. And a bunch of academics mostly are working on adding some robotic features to those pill cams so that they can deliver drugs and actually move through the intestine to the target site and take a video of a specific location like a tumor or a polyp inside the intestine. But nobody has developed a sampling capsule which can take samples of intestinal contents. So that's what we're aiming to do here. And so here's some of the requirements. Of course, it needs to be small enough to swallow. It needs to be biocompatible. So the journey is quite harsh through the very acidic contents in the stomach. And we want to make it from materials which won't affect the body or the microbes that we're collecting. We want this thing to be able to activate using the magnetic field robustly, blind, meaning even if we don't know where the capsule is, we'd like to be able to activate it. And we need to seal up tightly so we can get non-contaminated samples from a single location. And lastly, we'd like this to be a simple tool. So this is a bit of a contrast from the surgical tool application I was talking in the first half, where that surgical tool is quite a complex system, and it's designed for expert users in a well-outfitted environment. And the focus is on functionality and complexity is required there. But this tool we think we can design to be quite simple. And we'd like this to be simple, so that we can deploy it at scale, at low cost. But we'd also like this to be accessible by a wider population. So we don't want to design a tool which is only usable at premier Toronto hospitals, but something that we could mail as a sampling kit even to patients for doing studies, or down the road, hopefully as a diagnostic tool or even a treatment tool. So we want this thing to be simple. And so this is our sampling capsule details. And this work was primarily done by my former postdoc, Payman. And this is the view of one of our rough versions of our capsule. It has 2 magnets inside. And when we apply a field, this thing cracks open like an Easter egg. And you see there's a chamber inside that collects the sample of intestinal contents. And this thing is pretty simple. It's the same basic principle as the gripper I showed earlier, but this is just sort of folded up into a capsule shape. It has a hinge at the bottom, and we reinforce this thing with some carbon fiber, flexible carbon fibers throughout so that it could survive the journey. And here's a video of this thing activating. It's very simple compared to the other demonstrations I've shown. We apply a field, in this case, just by a big magnet, and the capsule orients and opens. You see it cracks open. When we remove the field, it snaps shut and seals tightly. And so we did a number of tests. These are in Ex-Vivo, actual pig intestines with intestinal contents in there. We did some tests. Will it seal? Will it leak? So this, we're running the capsule through sort of manually and testing. We don't want it to pick up sample at an undesired location. So we tested that, and we've shown that we can do that quite well. And second, can we activate it? So we did a similar series of tests in the lab, and then in these Ex-Vivo samples, can we put it to a specific location and apply the field? So we activate it, and then afterwards observe that we collected a sample. You can see the intestinal contents were collected into the capsule here. And so rather than using complex magnetic coils, for activation, we're using just a big permanent magnet. And the magnet fits into the palm of your hand. In the video here with the pigs that we did some trials with, live pigs, with our collaborators at University of Guelph, we put the magnet into a big box just to make it easier to handle and a little bit safer. And you see, we don't even know where the capsule is. We fed the capsule to the pigs, and we waited for a while, and then we just wave the magnet over it. And we've done a lot of study on how big does our external magnet have to be, and how far does it project the fields into the body. So based on the abdomen size, we can be ensured that it will open. And so we fed 9 capsules to 2 different pigs, and you can see here, all but one was able to collect a sample. And in this case, we sacrificed the pigs and showed that we can take samples. In this first trial, we were just sampling inside the stomach, and we plan further trials where we will have the capsules progress into the small intestine of the pigs, and we're going to hopefully this summer, if we're able to do some tests with COVID restrictions, we hope to be able to do a full sequence, passing the capsule completely through a pig and taking samples in a realistic way. Okay, so what's next for our capsule? We'd like to know where the capsule is. So ideally, we'd be able to sample precisely. And so I have some students, Xiaoyun and Daniel, working on ultrasound tracking. You see here just a video of our capsule opening. You can see it cracking open under ultrasound there. So this has some promise, and they're trying to apply some machine learning techniques to do automated or semi-automated detection of the capsule. We'd like our capsule to have some other bells and whistles, like multi-sampling, so one capsule taking multiple samples. And my student Priscilla is working on capsules that can activate themselves at the right time by using the different levels of acidity throughout the GI tract. So you'll have to stay tuned for that. And our plans, we're planning more animal trials, as I mentioned. And ultimately, in the coming years, we'd like to be able to do a study in humans, feeding these to people, and we'll be targeting inflammatory bowel disease as a study. Okay, so that concludes my talk. Just some final conclusions. This is a great space for innovation. As you see, my students and I are able to come up with lots of really fun and clever designs, and there's much room for and need for new innovative solutions. So this is a great and fun area to work in. There's a number of other applications that I didn't talk about here for drug delivery, taking biopsy samples, and even some that we're working on non-medical, like microfactories that can manipulate tiny components. And I'm often asked, how long is it till these are implemented? How long till these are doing surgeries? And of course, it depends on what device and procedure we're talking about. Something like our capsule, sampling capsule. We're doing this in animals now, and this is the basic function has been proven, and we think within a year even we could be feeding these to humans. There's a number of regulatory hurdles and technical hurdles before we get there, but that one's relatively close compared to our surgical tool project. Is a much more ambitious sci-fi kind of idea, and we've made a first prototype. So a timeline to implementation is really hard to say at this point, but animal trials will be coming soon, but in humans will be a while longer. So lastly, I'd like to thank my great lab group of talented, diverse group of students and postdocs who've done all the work that I showed here. And I put little blue arrows next to those who did the work that I showed in this presentation. But it's really a pleasure to work with these people. And I'm really fortunate to have such a great group of people. And so with that, I'm happy to take any questions.
Well, Eric, that was a marvelous presentation. I think it's very cool work. And I commend you, by the way, for acknowledging the various students, both at the end and during the presentation. I think as professors, it's important that I presume you've come up with many of these ideas, but it's most of the work, all of the work is done in the lab by really talented and passionate students, isn't it?
Yeah, that's right.
So to all of you listening, just a couple of things. We're getting questions both in the chat and in the Q&A, and you're welcome to put questions in there. I'm going to ask Eric some of these in just a minute, and we'll try to get to as many as we can. I also want to make people, just a reminder, make them aware that we'll finish this up at about 1 p.m. Toronto time. And then I believe you were provided instructions, and we'll put them back in the chat or the Q&A. But there'll be a little social event after 1 o'clock. I think Eric and perhaps some of his students, I'm not sure, will be going over to an app called GatherTown, which is more of a place. It'll be a place to have a little get together where people can talk and whatnot. And I think for as long as you're welcome to hop over there at 1 p.m. And then for as long as there's anything interesting happening, Eric and others will be there to further answer questions. So let me get to a few of the questions. I'm going to cherry pick these a little bit because I think we may not get to all of them. Just could you talk a little bit about companies you may be collaborating with and what the prospects for commercialization are? In other words, when does this go from being an academic project to something that surgeons and whatnot can buy and use?
Yeah, so our surgical tool project, as I said, this is a bold idea. It would really be quite a shift in how things are done, and there are a lot of technical challenges to solve first. So this far, we are not working with companies on a short-term commercialization path for that project. Although I think in the coming years, certain aspects of it, we really would like to involve companies and be thinking about that more seriously. Our sampling capsule project is more straightforward, and a number of companies we've been talking with, and no one that we're working with yet, but the companies are interested in drug delivery. There's an enormous commercial market for drug delivery using a similar kind of device, And so we are looking to partner and navigating the process of generating data from animal trials such that we can get regulatory approval to be working in humans is quite a process. And we would like to work with companies to do that, who are experienced in running trials and generating data and navigating that environment. So that's something in the near term, in the coming short months to years, we will be working with companies on that project. Yeah.
Great, thank you. A couple of people asked about the reusability of some of these tools. Now, I think the answer will depend on whether we're talking about the capsules and little grippers that are independent versus the surgical tools you spoke of. But can you just talk about, for example, the permanent magnets that you use? How permanent is permanent? In other words, are tools like that reusable? And to what extent are you considering reusing some of these things as opposed to them being kind of one-time use only devices?
Yeah, most surgical tools are reusable, and they're expensive, and they're masterfully machined metal mechanism components, and these are there's a strong case for reusing them. And I think the same thing would be true for ours. The magnetic materials, you've highlighted the most difficult part. For example, autoclaving these would be the ideal scenario. And there are magnetic materials that could go through that process. So, our prototypes that we're generating now are not sterilizable to that level, but I think that they would be able to, if we had a commercialized version of this tool, I envision that they would be reusable end effector actual tools on there. And the rest of the instrumentation also needs to be sterile for the environment, which for current surgical robot systems, that's actually, it's a challenge to do that. And often they put sterile bags over all the equipment around. And so something like that would likely be necessary in this case as well.
Right. Thank you, Eric. We've had a couple of people ask different questions about cameras on these devices. So, for the surgical tools, I'm sure there would be cameras, just as the Da Vinci device has cameras. But for the little independent devices, can one imagine, whether on the capsule or some of these actuators and these little grippers and whatnot, that one could put cameras so that one knew what one was gripping and grabbing and slicing?
Yeah. So our vision here is to have an endoscopic camera, and I'll just go back and you can see how this would be arranged in sort of the most logical configuration of this. You see on the top right here is the cylinder, which is inserted into the brain, called the trocar, which has multiple holes through it. And we insert our tools through two of those holes, and the third hole actually has the camera. So here's a view through that camera, the endoscopic camera, which is inserted right behind the tools. So it's kind of like you have your hands, if you look at my video here, I have your hands right in front of your face. And this viewpoint is not great. And so surgeons doing endoscopic procedures now work with this kind of viewpoint. The camera could also be inserted from a different hole. Multiple cameras, like anything is possible. And depending on the procedure, like surgeons do a lot of preoperative planning to figure out what exact tools are needed, what entry points, what viewpoints are needed for these kinds of endoscopic procedures, and would have multiple options for that. We'd like to be able to do things like use this camera view for doing feedback control. It's very challenging. It's not a good viewpoint. It's a messy environment as well.
I'm sure.
It's a big challenge in all of surgical robotics. It's a grand challenge going on now. Yeah. Right. Yeah. Our case is not unique.
I'll try to get to a couple more. So our own Professor Paul Milgram asked a question. So in addition to pinching and translating motions, in other words, is it feasible to generate rotational motions that you could use, for example, almost to cut tissue?
Yeah, that's a great question. Thanks, Paul. We can do that. So we can apply rotating fields, and we can rotate tools. That's actually quite easy to do. Doing it in a precise way, like if we wanted to use that to damage tissue, some of our friendly competitors around the world are doing things like using microrobots to drill out blood vessels, to drill out clogged blood vessels. And so this kind of thing is possible, and doing it in a precise way I think, we're not there yet, but it is possible. It's a good idea.
And I did have somebody ask, what about people with pacemakers? To what extent can you apply these magnetic fields to just anybody?
So as I said, if you don't have magnetic materials, iron, nickel, or cobalt in your body or in your pacemaker, like it wouldn't distort our actuation. So we could still drive our tools. Now, whether magnetic fields would affect a pacemaker, I don't think anyone with a pacemaker would ever be able to undergo any of these. Other implants may or may not, be able to if this was implemented. Yeah.
And I'll ask you one last question, and then I think we'll wrap it up. So Marshall Ma has just put this in the Q&A. So and did say thanks for the great talk, and true enough. He asks, have you thought about sensing rather than actuating with the magnetic field? Could you control multiple satellites at the same time? And he imagines this would be very useful for knowing, for providing more control to these little devices that you have in the body.
Yeah, sensing is a great attribute that we want. For a wireless tool, it's quite difficult. For these capsules, actually, they're big enough to put. We can put a microchip and batteries on board that capsule. It's a centimeter wide. People do that, and so people also put sensors on. So I think we could outfit ours with sensors as well. These surgical tools are smaller. We can run wires to them, and we plan to put sensors on the shaft of this tool, as I think I showed. Other like wireless sensors that can go into the body to measure oxygen concentrations in the gut or something like that. I think there's a lot of room for that. Microelectronic like integration with the instruments, I think, is the primary technical challenge for doing that kind of thing.
Well, you know what? We're a minute to 1 o'clock. I just thought, Eric, I want to thank you for just a marvelous presentation, and on behalf of the 100 or so people who are watching this, thank you both for the presentation and for the great work that you and your students are doing.